clinical things that we hear on normal day-to-day activities, but something that's we imagine to do, but has been done by great scientists who are also clinicians. It is indeed a privilege to request Professor Subramaniam to invite the first speaker to the dais.
Yeah, I would like to request Professor Mashur Seno to come to the dais, and I would like to introduce Professor Mashur Seno. Professor Mashur Seno completed his B. Engineering from the Department of Biophysical Engineering. He is a faculty member of Engineering Sciences and PhD from Osaka University, Japan. After serving various academic positions in the Faculty of Engineering, Okayama University, he is presently the Dean of Graduate School of Natural Science and Technology at the University of Okayama. He was a SAP member of the peer review committee for the LRI organization by the Science Task Force Japan and the Chemical Industry Association, Tokyo, Japan, in 2000, and also a visiting scientist of Lab of Tumor Immunology and Biology, NCI, NIH, MD, USA, between 2000 to 2001. Now, can I request now Professor Seno to give a lecture on cancer stem cells derived from mouse independent pluripotent stem cells? Yes, please.
Thank you, Chairman, for kind introduction. And thank you for the organizers, and especially Director Professor Abraham, for inviting me for the plenary session of the lecture. Today I'm talking about the cancer stem cells developed from the iPS cells. Actually, you all know that the iPS cells are available for the tissue regeneration, and all people are expecting the iPS cells available for regeneration therapy because iPS cells has the multiple differentiation potential depending on the microenvironment to introduce progenitor cells, which is destined to reach matured cell types. And many people are worrying about the tumorigenic situation of the iPS cells, but considering the multipotential of the iPS cells, why not, I thought, the cancer stem cells could be introduced destined to the cancer cells. So the question was that, what kind of cancer niche is considered situation to induce? So we made it available to induce the cancer stem cells from iPS cells developed by Professor Yamanaka, which is published in Nature in 2007. And these iPS cells are very easy to distinguish the differentiated and the non-differentiated situation, because the GFP gene is integrated under the nanopromoter to produce the iPS cells, introducing the Yamanaka factors. For the cancer niche, cancerous niche, we consider about the using conditioned media from the cancer cell lines. And two conditions we introduced. One is conditioned media, and one is co-culture together with the cancer cell lines. And the keeping the culture for 4 weeks, both, and then transplanted to the nude mice to confirm that the tumorigenic, to confirm if they are forming malignant tumors. Usually, iPS cells are transplanted in nude mice to find out normal tissues to forming teratoma. Actually, teratoma contains normal tissues such as squamous epithelial cartilage, neurons, neuroglia, and this is lymphatic tissues. These are every tissue is normal. You know that teratoma is benign tumor. No malignant tissues are found when you transplant iPS cells into nude mice. So, what kind of cancer cell type tissue cell phenotype is found? This is the malignant tissue produced by the transplantation of the iPS cells treated with conditioned media from Lewis lung carcinoma, LLC cells. The section shows that the malignant phenotype with gland epithelial hyperplasia, this is, and high nuclear to cytoplasmic ratio and severe nuclear atypia. Multiple nucleotide mitotic figures shown in red arrows, and extensive angiogenesis. And in blood vessels, you can find micro metastases here. So that all these factors are, considering all these factors, these tumors are judged to be malignant. So quite different from the normal tissues displayed in teratoma. And we tested 4 different cell lines to confirm that the phenomenon is not specific for the Lewis lung carcinoma conditioned media. This is Lewis lung carcinoma. This is TRAT carcinoma. This is melanoma, and this is mammary carcinoma. And interestingly, conditioned media treatment showed almost all the malignant, almost all the cases induced the malignant tumor in nude mice. And on the other hand, the co-culture system did not show any tumorigenesis. This is the case. The Lewis lung carcinoma co-culture. There is no progeny of the iPS cells, so that we couldn't transplant the cells into nude mice. These cells from teratoma and melanoma did not show any tumorigenicity. And this is only the exception when the co-culture with mammary carcinoma showed the malignant tumors. So that this is the key point to show the conditioned media as the cancerous niche when you convert the iPS cells to the malignant phenotype. We confirmed that the GFP expression ratio in the resultant cells. This is iPS cells cultured without leaf and meth, these are feeder cells. And almost 24% of cells are positive for GFP. And this is the case of mammary carcinoma co-culture. This is melanoma conditioned media treatment. This is a teratoma conditioned media treatment. This is Lewis lung carcinoma conditioned media treatment. The GFP positive ratio is depending. You can find different, so that it is not. You cannot tell GFP positive ratio is responsible for the tumorigenicity of the iPS cells treated with the cancerous niche. And some papers are reporting that the cancer stem cell-like cells are converting to the endothelial phenotype, so that we evaluated the tube formation assay of matrigel using the resultant cells from iPS cells. This is in vitro experiment. iPS cells does not show the tube formation in the presence of collagen type 4, but the mammary carcinoma co-culture cells and throat carcinoma co-culture cells show the weak tube formation and clearly different from these cells. Luis lung carcinoma condition media treated iPS cells showed a strong, significant tube formation. On the other hand, melanoma condition media treatment did not show any tube formation in vitro. And this is impressive because the tumor formed in nude mice using these cells showed extensive angiogenesis. And this tube, this was shown by the immunofluorescence against the CD31, which is the marker of the anterior cells. And this is GFP expression in the tube formation, and so that the overlay of these pictures are shown here. And red part, and green part, and yellow part, each of them are observed in this figure, showing that this is the DAPI staining, so that the live cells are here, so that there are cells that is not positive for both GFP and CD31, so that we can judge that the iPS cells are differentiated into GFP positive and CD31 positive cells, and GFP1 positive and CD31 negative. And again, this is a GFP negative and CD31 positive. This is the N0 phenotype, and still GFP1 negative and CD31 negative. So we can confirm that at least four different phenotype the cells are composed in this tube-like formation. That means the heterogeneous structure. And this is the appearance of the tumor formed in the nude mice. You can find the blood vessels are coming into the tumor. And we can find that the small blood vessels of angiogenesis surrounding the tumor are appearing. And this is the gadolinium-loaded injection of the tumor, observing the MRI angiography of vasculogenesis. And just after the injection, you can find the thick gadolinium positive blood vessels, big blood vessels can find immediately. And 2 hours after, you can find the inside of the tumor is observable, showing that implying that the tumors forming the blood vessels inside, differentiating into the endothelial cells, and probably the host blood vessels are make junction together with the blood vessels in the tumors, so that tumor angiogenesis will be the mechanism of the tumor angiogenesis should be cleared using this type of observations. And this is the section from the tumor. This is the normal iPS cells transplantation. I mean, this is teratoma. This is malignant tumor formed using the Lewis-Lung carcinoma conditioned media treatment. And you can find the epithelial cells stained with anti-cytokeratin antibody. And this is also the developed epithelium stained with, strongly stained with the cytokeratin antibody. And also the stromal tissues in the teratoma is negative for anti-GFP positive antibody, showing that these stroma tissues are composed of differentiated cells. On the other hand, the stroma tissue in the malignant tumor shows that partly GFP positive, partly GFP negative, showing that differentiation of the cells are composing the stroma tissues of the malignant tissue. From this result, we can see that the GF positive and cytokeratin negative cells and the GFP negative and cytokeratin negative composing the stroma together with the epithelium, epidermis, as GFP negative and cytokeratin positive. So we can find that there's at least 3 different cell phenotypes composing this tumor tissue that is heterogeneous. And from the tumor tissue, we can isolate the primary culture of the cells with green fluorescence together with the fibroblastic cells. These cells can be cultured as a spheroid, as a suspension cultures, which is showing that GFP positive, so that these spheroids are composed of the undifferentiated cells, depicting the self-renewal. And when the adhesive culture is carried out, these spheroid cells are developing into the fibroblastic type of cells without GFP negative phenotype, showing that these cells are differentiated from the iPS undifferentiated state. So we can judge that these cells are cancer stem cells equipped with the self-renewal potential and the differentiation potential. And the spheroid cells are nanopositive when stained with nano antibody, and also OCT34 antibody. This is positive for these factors. This is DAPI staining for control. And again, the primary cultured cells are transplanted into nude mice. So you can find the phenotype of the tumor tissues are the same character of the first tissue. Cytokeratin positive for the epidermis, and GFP positive and negative part in the stromal tissues, and highly extensive angiogenesis, and stained with the anti-CD31 in the tissues. This is when this is the metastasis experiment. The cells are injected from tail vein to find out the metastasis of lung, and this is heart, and this is normal lung tissue, and these nodules are metastasized iPS-derived cancer stem cells. And you can find the tumor tissue here together with the lung tissue, and here this is a tumor tissue with the extensive angiogenesis, and this is the tumor tissue stained with anti-GFP, showing that again that the GFP positive, partly GFP positive and partly GFP negative, showing that heterogeneous component still exist present in the tissue. And this is the summary of the iPS-derived cancer stem cells, showing that self-renewal as a spheroid and differentiating NCL cells and producing malignant cells, malignant tissue in nude mice. And we can find another example isolating microvesicles from the Lewis lung carcinoma condition media and treated with the same condition for weeks of iPS cells. And this is another phenotype of the differentiation into adipocytes and differentiation as well as differentiation into vascular endothelial cells. And in this case, spheroid cells appeared GFP positive and negative. We are now confirming that this is forming malignant tumors in nude mice at this moment. And this is the tumor tissue produced using the cancer stem-like cells and showing that adipocyte character stained with the all red staining. And so that we are judging this lipid sarcoma here. This was adenocarcinoma for the example 1. So that we can get the different tumor phenotype of cancer stem cells, starting from the iPS cells, so that normal niche would testing the iPS cells or embryonic stem cells into the normal cells by way of the progenitor cells. But when the cancerous niche is encounter these cells, once CSC is produced, the progenitor of the cancer cells will be produced, and progenies will provide various phenotype of cancer cells. And considering the heterogeneous of malignant tumor, these components are composing that one malignant tumor, and displays the heterogeneity of cancer, and providing the metastasis, invasion, and angiogenesis, and which will result in the poor prognosis of cancer. Still, we are saying that 5-year survival rate and remission, recurrence, and so on, but no complete cure is not expected at this moment. And so an insight into cancer stem cell from this model, heterogeneity of cancer tissue is proposed. Cancer stem cells variously exhibit differentiated and undifferentiated phenotypes. And mixed phenotype of cancer cells appears the character of heterogeneity. These are the collaborators of laboratory, and Lin Chen performed almost all the part of the experiments, and these are the department of PhD, staff of our laboratory here, PhD, and Dr. Srija Sekar is our graduate who received the PhD this September, and she is now in this meeting now. And he is from also India, and was a graduate student in my lab, and now working in NCI in Bethesda. And these two are good advisors from the Northwestern University and NCI Fredericton. And Professor Li Fu is a professor of the Lin Chen, and Koichi Igarashi is currently provided the experiment of the MRI angiography. This is our campus of the Okayama University. This is the graduate school of natural science and technology, and this is the faculty of engineering. This is the faculty of sciences, and together with these faculties, we are conducting the natural science and technologies research and education. And in April, we can enjoy the cherry blossom, and in autumn, we have ginkgo trees, so that quite different colors we can enjoy in autumn and spring. It's a very beautiful scenery here in Okayama University. And we are the students in our laboratory. And thank you for your attention.
You have 5 minutes, you can ask any questions. Now, as you all of you know, there is a lot of excitement after the Japanese group, Yamanaka et al., have come forward with the suggestion that cells can be made again into pluripotent cells. Ordinary mature cells can be made into pluripotent cells, can be used for the various regenerative conditions. And this amount of excitement which caused him to have the Nobel Prize also, he has been getting again nowadays faded away because of the one that is circumvented with the ethnicity and other things. The problem of the tumorigenicity is still existing. I would like to put the question before I ask the audience, what do you think, Dr. Senu? What the factors are really playing the tumorigenicity? Is it the genetic material that is injected, or the genetic material that is given like OCT434 or SAX2, or other factors like KLF4? People believe the material that is being given, the genetic material given, is going to play a major role. When compared to the earlier given OCT34, the recently given KLF4 is more tumorigenic. Do you agree?
Mm-hmm. Yeah.
Do you agree what I ask you, that is KLF4 is more for tumorigenicity than other genetic material?
Well, that is a good question. First of all, we are now investigating the factors which will induce the iPS cells into converting into the cancer stem cell-like phenotype. And conditioned media contains many factors, as you know, and we are now separating the exosomes together with microsomes derived from the cancer cells. And as I showed you, that the exosomes and the microvesicles inducing the lipidosarcoma quite different from adenocarcinoma, which is treated with conditioned media. And then, I didn't show the data, but the conditioned media without exosomes and microvesicles shows different type of cancer stem cell-like tissues, so that the many kinds of factors could be inducing the cancer stem cell-like cells, and so that we should investigate much more to identify what critical factors will be included in the conditioned media. And as for the induction of the transcription factors, actually Yamanaka factors when transduced into the cells, the introduced genes are not keep expression. You know, they appear that only in triggering the expression of endogenous genes, and so that the KLF4, including another SOX, MYC, or endogenous genes are triggered. And when compared with the KLF4, in this case, KLF4 is suppressed in our first observation. So that the KLF4, in this case, KLF4 is acting just like two more suppressing factors. So that there are, because it is a reason because there are some different papers showing that KLF4 is acting tumor suppressing or sometimes tumor activating. So that these factors are determining the situation differently. So the time point of the induction of cells are critically different, and so that the results are different results are obtained, probably, I think.
Anybody from the audience?
Thank you for a wonderful presentation. Sir, you are told in co-culture there is no cancer cell growth, but due to cell-to-cell inhibition. But because of paracrine effect, there can be a change, no, sir? Paracrine effect, between the cell-to-cell paracrine effect. You are told in the co-culture there is no cancer cell growth.
Yes.
There is no, but because of cell-to-cell inhibition. But due to paracrine, between the cells and cells, we have paracrine effect in the cancer growth. Can there be a change? No, you are not found a cancer cells. Absolutely no cancer cells in the.
So your question is why co-culture did not show malignant? I think that the co-culture shows both communication of cells. If you make co-culture together with iPS cells, cancer cells, iPS cells are sending signals to cancer cells, so that cancer cells should be responding to the signal to send the message to iPS cells. There should be the communication between the cells. This is the key point, because conditioned media sends only the signals from cancer cells to iPS cells. iPS cells should be responding to cancer cells and sending some message, but there is no responder here, only in conditioned media. So that this is the key point, I think. So that some people are showing the reports that the cancer cell treatment using stem cells. So that in this case, stem cells is sending the messages to cancer cells, so that the absence of the message is the key point of the producing CSC cells, I think.
Yeah, sorry for want of time.